like I said, I grew up in Pacifica and I currently live in Berkeley, but I've jumped around a lot over the past decade and a half almost. That's a long time. But anyway, what I want to talk to you all about is sort of my experience in in academia and going through my education and ending up where I'm at today. So the title of my talk here is really turning that degree in marine biology into a career in biotech. Oh, and what I'm showing here is actually a green flash that we captured off Station Aloha, which is 60 miles north of Hawaii. I'll talk about that in a little bit more, but it's kind of cool. When the sun goes down, you can see it actually change color because the light's refracted from the atmosphere. They happen, they exist. It's not just a thing in Pirates of the Caribbean. But yeah, so you get to see some really cool stuff when you spend a career in the field. So a little bit of my background. I have a bachelor's in marine biology from UCSC, in which I'll talk about in a second. I didn't really plan on going to college. It just kind of was a means to play water polo. From there, I was a guest investigator at Woods Hole and MIT. From there, I became a biogeochemical specialist at the University of Hawaii. From there, I did my Ph.D. in cellular and molecular biology at Georgia Tech. Then I became a metagenomics scientist at a company called Dovetail Genomics. And let's see, we're getting chats here. Okay, got it. And then my current role is scientific affairs lead at Dovetail Genomics. So I'll walk you through a little bit of each one of these steps and kind of how I ended up in my career, just by kind of riding the wave a little bit. So I never really wanted to go to college. I wanted to go to college to move out of the house and to play NCAA water polo. And you can't really see, but if I'm standing in front of you, I'm like 5'9" on a good day. And so I knew I was going to be playing D1, but I knew UC Santa Cruz allowed walk-ons, and I had gotten into the university. So I had really gone to college to play water polo. The only career path I was somewhat interested in was opening an aquarium shop. At some point in time, I built aquariums and used to grow coral and all that fun stuff. So that was really the only trajectory that I saw for myself when I started college. And because of that, the first 2 years didn't go so well. I barely passed Gen Chem. I almost had to retake genetics. Like, I just barely skated by here due to some time having a lot more fun than going to class. I ended up having to retake Calculus 3. So my GPA at the end of the first 2 years was a solid 2.2. It was good enough to allow me to play water polo, but not really good by any other means in order to move forward with any career. It was sort of at this time that I realized that 5'9" and 180 pounds at that time was not a sort of the makings of a lifetime career in water polo. And so I kind of buckled down in the last few years of my undergrad and bolstered my GPA to a 3.9 during that time. So I finished with a GPA of 3.18, which isn't outstanding by any means, but it's a lot better than 2.2. But I ended up graduating with honors for my work on my senior thesis, which I'll talk about here in a second. But what really allowed me to really turn around my focus in academia was getting involved in microbiology. So in my third year, I took this class in marine microbial ecology. And if you know anything about fish tanks, it's basically fish tanks are a bioreactor for microbes that move nitrogen. So I had a pretty good understanding of the nitrogen cycle and the bacteria that help move around those chemicals. But at very much a cursory level and at very much a hobby level. So I'm just sort of showing here at the bottom what the total nitrogen is in an aquarium and what forms it takes shape in, and how you actually remove those through things like water changes and the bacteria that chew up ammonia and nitrite. So I ended up taking this class with this professor named John Zare, and him and I got along great. There was, I think, 10 people in the class. 8 of us showed up for every single class, and there was only 2 of us that actually asked questions and talked. And at the end of the class, I handed in my final, and he offered me a spot in his lab, literally just washing dishes. I was a dish boy for a lab. So I was, I learned how to do sterile cleaning. I learned how to autoclave. I learned how to just basically stock shelves. I was just kind of the errand boy for the lab, checking freezers, making sure that, there was still ice in them, that the -80s were at -80 and all that fun stuff. So the longer and longer I stayed there, though, after you kind of put in your few months of just kind of being the whipping boy, I was offered experiences in how to do DNA extractions, how to clone, how to do PCR, and really just basic laboratory techniques. And then from there, too, I was invited to go on a few day cruises out in Monterey Bay. Now, what that culminated into was very much at the time an unwanted thesis. I still was really not quite sure that I wanted to do any more work in this field, but I knew that I still liked microbes and I liked fish tanks. So my advisor there, John Zare, had asked me to team up with Kendra Turk, who I'm showing up here, to help her do some work in the Monterey Bay, looking at a particular gene and some cyanobacteria. Because I had all the skill sets, they just said, here are the samples that need to be extracted. Here's the primers for the PCR, go off and run with it and turn it into a thesis. And I was like, that's a lot of work. I'm not sure if I really want to spend that much time. And they're like, no, if you're going to work for us, this is what you're going to need to be doing. So I did that work, and I actually found a new strain of the Cyanobacteria Synechococcus in the Monterey Bay, which we had isolated from this station here. And so we're pointing arrows to the clones that we had identified that actually didn't group with any of the other known strains of this particular cyanobacteria. And so that was cool. It was an interesting piece of work for me. It was a good experience that all those sort of lab techniques that I had been shown just basically as a technician were actually useful in answering real biological questions. So I'm showing the chemical pathway here, and the NTCA gene that we were looking at really mediates the GOGAT system, which tells cells to either pull in nitrate or ammonia for biosynthesis or for energy turnover. But anyway, it was a really good experience for me. It showed me that the skill sets that I had learned as a dishwasher, basically, for the lab had actually translated into some meaningful science. So from there, when I graduated, I didn't really know what I was going to do, but my lab knew exactly what I was going to do, which is kind of cool. They had introduced me to this professor named Dan Rapeda at MIT and Woods Hole Oceanographic University. And this professor works very much in geochemistry. Now, again, I had barely passed basic chem. I wasn't probably the best candidate for this role, but what they wanted me to do was quantify organic molecules out of seawater. Well, if you think about it, DNA is an organic molecule, right? So if I can do things like PCR and qPCR, that's manipulating organic molecules out of a probably a slightly more complex matrix than just seawater, that I should be able to do similar work at a just kind of hardcore organic chemistry level, rather than thinking so much about the molecular biology and the microbiology. So the project that I worked on for Dan was this aerobic production of methane, which is interesting because we think methane as a product of anaerobic processes. So like mud flats, or if you dig deep enough, you start kind of smelling sulfur, and then below that you get methane deposits, right? So we really do think about that as an anaerobic process where there's no oxygen. But that's actually not the case. There's a layer in the ocean that actually is quite enriched in methane that's close to the surface. And we're finding out that microbes that are starved of phosphorus start moving to chew up organic carbon phosphorus bonds. And in the process of cleaving the phosphorus off for things like DNA, you have to have a phosphorus layer for your backbone, for proteins, your cell walls, all that fun stuff. So in order to gain that phosphorus, they were cleaving at that carbon phosphorus bond. And in the process, they were actually producing methane, ethylene, and phosphate and all that fun stuff. And so this phosphate was then going in to feed a much more complex world of microbes at the surface than what we had initially thought. So my job there was to learn how to do HPLC mass spec work. So I got pretty good at managing the mass spec and characterizing organic phosphoruses out of seawater. So just by 6 months after leaving my undergrad, I had developed a whole new suite of techniques in organic chemistry, which was pretty cool. Then what happened was the grant that I was being funded on by this professor ran out. It didn't get refunded. He didn't really have money to keep me on board. But what he did know was there was this professor, David Carl, out at University of Hawaii, who was running a field-based operation where we would spend 3 months out at sea sampling every 4 hours for that 3 months. So he was like, they're looking for people. We need folks that have experience. You have a little bit of sea experience, would probably be good for you to get more. You should go. So I said, okay. So I packed up my bags, stopped off at my folks' house in Pacifica, dropped some stuff off, and then flew out to Hawaii. I was there for a week or two helping prepare, and then we took this ship. This is the Kila Moana, and we went out to Station Aloha, and we were dropping sampling equipment, I kid you not, every 4 hours on the dot for 3 months. It was a long, long time out at sea. I made some really good lifelong friends though. And it was, like I said, it was a killer experience. And the cool thing is because now I've had this lab-based and then field-based experience, I was actually pretty well known throughout the world of marine microbial ecology, just because I had been exposed to so many people, being not just at UC Santa Cruz, but being at Woods Hole, being at MIT, and then again at Hawaii. These were big hotspots for people doing microbial oceanography. So I at least became well known within that circle. So when I was starting to look and say like, okay, the work that I had done previously between Hawaii and Woods Hole was probably good enough for a master's, even though, I'm not actually in those classes. I learned a ton. And it gained a lot of experience and was helping with producing some really cool primary research work. So it's like, I should probably start thinking about, doing my PhD, or at least go to grad school for this. And, I was joking around, and one of the guys that I was with at the time, he said, oh yeah, my postdoc just left, and he was a big collaborator with David Carl. And so he said, yeah, my postdoc just left. He's looking for grad students. Why don't I have him give you a call? And so the next day I got a call from a professor at Georgia Tech saying, hey, we'd love to have you down here. And, we just on our first call started brainstorming about what the PhD would actually look like. And so my background at UC Santa Cruz was nitrogen cycling and microbiology. And then my background at Woods Hole was phosphorus cycling and methane cycling in chemical experiments and rate experiments from the ocean. And then my experience in Hawaii was geared towards nitrogen chemistry and, again, field experiments. So it kind of made sense that we would couple things that I had really worked on to open up a whole new avenue of discovery in the ocean. So we had, before I'd even really started there, it was that the idea was that I'd come to Georgia Tech to start investigating microbes that couple nitrogen utilization along with methane utilization in the oceans. And that's where my PhD advisor, Frank Stewart, comes into play. He works in these systems that are anaerobic. So there are naturally occurring regions in our oceans that basically lose all oxygen. So one of the more famous ones is the Gulf of Mexico, where you get massive runoff from the Mississippi and Apalachian Basin, and fuel algae growth. That algae dies, sinks to the bottom. The bottom waters become hypoxic, and that has big implications for fisheries and the shrimperies that are out there. But this is a process that actually occurs naturally as well. So off the western boundaries of continents, you see one here, and then off the coast of Namibia, as well as in the Indian Ocean, there are regions where upwelling occurs naturally. So, in Pacifica, being right on the coast, the springs, you see all, like if you drive down the coast, drive towards Santa Cruz, you see all these massive kelp growths that kind of come up to the surface. And that's all driven by these nutrient-rich waters that are moving from the bottom of the ocean up to the surface, that are fueling phytoplankton and fueling big kelp forest, right? Well, the same principle applies. All of that, those nutrients come to the top, fuel phytoplankton, and then as they die, the microbes use the oxygen in the water to chew up all of that sugar that's been converted from CO2 by phytoplankton. So as you move down in the water column, these places just become stripped of oxygen. They're absolutely anaerobic. And it's really cool. NASA funds a lot of research in these areas because they think of it as a non-Earth analog system for how, like, maybe sulfur or methane based life would exist or could exist. So there is quite a bit of money in actually looking at these regions as places for what life on other planets might look like, as well as, resources. What resources are being produced down in these regions that we don't actually know about. So really the goal of my PhD was to do metagenomics and to figure out what microbes are down there doing what. And so if we look at what genomics is compared to metagenomics, genomics is taking the puzzle piece that is random shotgun sequence data and turning
Cory, can I interrupt you on one thing? You're doing great.
Absolutely.
What I'd like to do is kind of, you were talking a little bit about how basically you got all your positions by knowing somebody in your previous position. So yeah. How do you do that ask of, can you find me a job, or what did you do that kind of helped you get into those positions? How do you form those relationships with people to kind of help get you that next job? What did you do?
Yeah, that's a great question. So I do have a few slides at the end, but the punchline is, one, you have to talk. You have to talk to people. That you can't really sit quietly in a corner and expect things to happen. The next one is ask questions. So in classrooms or in lab meetings, if you don't understand something, speak up, right? People do want to teach you things. And so once you've established yourself as somebody who wants to learn something and is willing to put in the time, energy, and effort to capitalize on somebody who's imparting that knowledge, time, energy, and effort, they're willing to go to bat for you. They are willing to help you out. So, in lab meetings, I remember being an undergrad in these lab meetings with only grad students where they're talking about, sequencing costs. Like, what's the most important sequencing platform? And I'm just raising my hand going, what are the sequencing platforms? I don't know. Somebody tell me. And of course, everybody else in the room knew, but I didn't know, but I wanted to know. So first off, showing that you're willing and able to be like, teach me something and I'll use it. And then the second thing is to be really helpful. Just try to help. Ask people how you can help them. Nobody's going to turn down help. So the more you make yourself available in that aspect, you gain skill sets, and then you can show that, okay, if you teach me something, if you train me in this, I'm going to help you out. This is going to benefit you. And so the more you become a person that somebody can rely on, right, that relationship becomes much more than mentee and mentor. It becomes a collaborator. You are somebody who, your advisors or your superiors can lean on and know that you're going to produce, right? So, speaking up, being willing to accept responsibilities and offering help, those things go a long, long way. And then they get invested in you. They have spent many, many hours of their life training you, and they don't want to see that training go to waste, right? And they can see a valuable ally in you as a colleague, as a future colleague, rather than just somebody I taught a number of years ago, right? And so that's what I've actually been told by my professors and my supervisors. Is just like, we want to develop a future colleague. We don't want to just develop a better student. We want to develop somebody who we can work with in the future. And so by doing those things that really, one, establish yourself as somebody who can handle responsibility and not to shy away from it, and then also to produce after you've been trained on something, those two things go a long way, because they'll see a colleague in you. They won't just see a student. They'll see someone who in the future they go, oh, I can reach out to them if I have a question, because they're going to learn more from other people and become a much more well-rounded person in sciences in general. And so when it comes time to, hey, grants are running out or I'm getting ready to graduate, it becomes part of the natural conversation. You're already talking to them about your day-to-day workflow form, right? Or your week-to-week workflow form. And you say, hey, in 2 weeks, I don't know what we're going to do here. Or in 2 months, I don't know what comes next. What do you think comes next? What do you see pathways forward for me? Here are some that I think could be useful. Here are some that I'm not so sure about. What's your experience? And a lot of times these things come up naturally. They'll ask you what you think you want to do and what you have coming up next. It's that dreaded question, well, what are you doing after you graduate? Well, those are really good people to go, I don't know. What do you think? Help me. And so by opening those kind of chains of communication early on and show that you're willing to help them, they are more than willing to help you when it comes to that time. Because everybody kind of knows everybody. The higher up you go in your education, or even in your field, even in your whatever work you decide to go into, there become fewer and fewer people that become either experts or that well known in those regions. And so at some point, the more and more you're around, the more and more everybody knows everybody. So if you can link into somebody that's that connected, and just be helpful, they're going to be helpful in return.
Thanks, Cory. That's great.
Yeah. So I guess that's one thing that I'd kind of like to impart on you all moving forward, is that, the reason that I was putting these images up of these, the people that I've been working closely with over the years, and it'll continue through most of my slides, is that none of this is done by yourself. This is all done with help from other people. And being collaborative and being open to those sort of lines of communication are really critical in driving your career forward. And no matter what you do, even if you want to go off and get a Ph.D. or if you just want to get a job at the end of the day, right? Being helpful and supportive of people, even if it's not in your primary nature, pays off big time in the long run. So those are things that I hope you take away from this, and that's why I'm calling out the people that I've worked with.
Great, thank you.
Yeah, no problem. So just kind of briefly, my job in grad school was to move from genomics, which is taking a single genome and piecing it together to what you would think of as a completed genome, to taking many genomes that are in the seawater, they're just living in seawater, and trying to complete what the puzzle looks like from that. And you're not going to sequence every single microbe that's in the ocean. So in 1 mil, just 1 mil of seawater, there are about 1 million cells. Well, 1 to 10 million cells, depending on where you are on the planet. And there are between 10,000 to hundreds of thousands of species within that 1 mil of seawater. So the idea is to figure out of that kind of hodgepodge soup, once you do your DNA extraction, what are the genomes that are there? How complete can you make them? And then what can you determine as to what they do? And so in light of trying to figure out what microbes use methane here, we found that somebody had recently discovered this really wonky strain of bacteria that has this really unique sort of octagon shape here, but really they do this absurd chemical reaction where they take nitrite and they take methane here, and they are able to, inside of their cell with no oxygen, break down the nitrite into nitrogen gas and oxygen gas and do an oxidative process inside of their own cell. And so it was discovered that these things exist, but we don't know exactly where they live. And we thought, well, nitrogen and methane both live in these anaerobic conditions, which those oxygen minimum zones were. And sure enough, they do. So we teamed up with some really kick-ass chemists. There's no other way to describe them. They are awesome human beings. Both Antrip and Laura Bristow, who I'm still publishing papers with Laura from my grad school work. So we went to these sites and we sampled for both the microbiology and the molecular biology and the chemistry that occurs there. And our hypothesis was, because these oxygen minimum zones are enriched in nitrite and enriched in methane, that these regions should harbor these bacteria. And they do. So here, if we're looking at the inputs to this metabolism, nitrite and methane, and we look at the methane and nitrite concentrations as we move down through the water column, we see that methane is indeed enriched in these anaerobic sites. We see that nitrite is indeed enriched, and the NC10 bacteria that does this metabolism peaks right at the interface of these two chemical peaks, which is really cool, because that's kind of how you expect microbial ecology to work. Is they're not going to live only where one substrate is or the other. They're going to live somewhere in between, where there's enough of each substrate in the reaction to actually allow them to live. And so we did this here off of Manzanillo, Mexico, and then we went down to Costa Rica and did the exact same experiment, and you kind of get the same pattern where the bacteria are enriched where the two substrates co-occur. So that was one of the papers that I had published. I forgot there were slides there. But one of the other things that we had found in doing that work was that in the Gulf of, or in Costa Rica here, that wasn't the only microbe that actually carried out that process. So we did some more work and figured out that there was a new cell type, this blue one here, that carried out a very similar process in a completely different type of microbe than that NC10 group. So that kind of rounded out my PhD. I'll skip over that. That's a lot of chemistry stuff. But that collaborative spirit that had been imbibed in me for throughout the course of my training actually led to many publications. So here's just actually subsampling of all the papers that I was on during grad school. Since leaving grad school, I've gotten another 8. We just submitted another paper 2 weeks ago from my grad school work. So I have tons of projects just by saying, yeah, that sounds cool. Let's give that a shot. That's a good idea. Let's try that. And so it was one of these things where publishing that much in grad school actually made me be able to jump into the workforce. So this was probably moving from my PhD into biotech was something that I actually didn't rely on anybody to make those connections for me. This is the first time actually in my career where I've been like, I need to go find something. I don't know anybody that works in biotech. You see a lot of people in the environmental research world, they don't really produce a lot of people that end up going into the biotech route. They kind of train people to stay either in academia or stay in environmental type sciences and jobs. So this was one case where I had to go reach out on my own, and having that level of publications and that level of collaborative spirit really caught people's eye when I was sending out my resumes. And actually, the person who hired me on at Dovetail Genomics actually told me, we hired you because of your publication record. You know, you've shown that over a short period of time that you can produce work, right? So that tells us that you do your job, right? If you're not, producing a lot, there's some questions about maybe you should go off and postdoc and learn a little bit more before joining the workforce. But because I was publishing a lot and had so many collaborations, they knew that I was going to work well in their team. And so just
well, Cory, just a couple of questions. So number one, you're already a little bit older. You didn't go directly from your undergraduate to graduate school. You'd worked a little bit of time in there as well,
correct.
right? So you're a little bit more mature in doing that. The second thing is, what was really kind of interesting before is, could these organisms be used as a way to kind of scrub out methane from the ocean to reduce our methane production, or?
Oh yeah, so they currently do, right? That's not something that you could try to actively engage them to work on, but so the thing is when you start removing oxygen from redox chemistry, your delta G starts dropping really, really fast. So we know that one of the biggest delta Gs in the biological world is sugar and oxygen, right? That's why our bodies are so attuned, and the mitochondria in our bodies are so attuned to take in the byproduct of that sugar oxygen redox reaction, right? So as you start stripping oxygen away from that redox reaction and start replacing it with nitrate and nitrite, or even oxidized metals, your energy yield from that reaction starts tanking very, very quickly. So can they do it? Absolutely, they can. But the more effective way would be to use aerobic methanotrophs as opposed to anaerobic methanotrophs to do that. Okay. So yeah, I mean that's good.
Yeah. No. Okay. Yeah.
But it's one of those things too. You never really know what it is you're going to find when you you set a hypothesis when you go out into the field. But then, like I was saying, like there's so many species in seawater, you never really know what it is you're going to find. So the sequence data that I had produced during the course of that publication has now been cited over 80 times. It's being used by other people to make other discoveries. So you never really know what it is that you're actually going to find down there and what could be useful and what's not. And a lot of times you don't know what's useful until, decades later, you go back and actually say, oh, this is actually the world we live in now. That's actually a useful, organism, or that's a useful chemical reaction. That's why most Nobel Prize winners are like 70 by the time they get it. Get rewarded, right? The discovery that they made in like 1960 is now being only recognized as that influential, right? So moving on to how I joined the workforce. So the company Dovetail Genomics, they do this what's basically called 3D genomics. We know just at a very high level, your DNA is not in a straight line in your cell. So human DNA is about 3 meters long. And that 3 meter long copy has to fit within inside every single nuclei in your body. And so in order for that your genome to be fit into that cell, it has to be folded up in along itself many, many times to be ordered to fit inside every single nuclei. And so one of the things that we found is how your genome is actually folded on itself determines what genes are expressed in that process. So it's a very hierarchical thing. You could zoom out really far. If you were, like, say, think about a microscope section of a nuclei, you can zoom in, you can see chromosome territories, you can see compartments of inactive and active DNA, you can look in and see clusters of linked function genes, and then you can actually see the anatomy of a gene where an enhancer and promoter are brought together, and then our pole 2, which is the thing that carries out the central dogma of biology, which is turning DNA into protein, allows to happen. So this company does 3D genomics. They have a method in which DNA is cut, and then you have all these free ends floating, and you tell it to bind and link to the DNA that's closest to it, and in many cases, that's not in linear space, it's in 3-dimensional space that it finds its closest DNA buddy. And so what you get are these sequence reads that map to, so for example, each pixel here represents a read pair that maps to some distance away. So read 1 would map here, read 2 would map there. So it's a way for us to investigate how your genome is folded in on itself. This is actually really useful in genome assembly because you can say, okay, within one of these chromosomes, we see far more contacts than we see across chromosomes. So all of these pieces of DNA belong to one chromosome. And so the idea was that you could do this with metagenomic samples, right? If you cross-link all the DNA with inside a cell after you do your DNA extraction, you'll have these chimeric reads that only occur within a cell as opposed to across cells. Long story short was that I made this method work, and then the commercial and marketing team said there wasn't a big enough market, so they dropped the product. And from there, I did a number of other things. I worked on developing, we were offered to buy a few products by Illumina, which is the biggest sequencing company on our planet. And they asked me to validate some of their protocols to see if they were actually worth buying. I had done that. We had decided that we were going to make an offer on one, and then a new technology came out that looked better, so we didn't buy that. And so I started playing around with that new technology called Oxford Nanopore, which is some really trippy stuff. If you have it up second, you should Google Oxford Nanopore sequencing. They do some really, really out of this world stuff when it comes to sequencing technologies. But anyway, so I worked to develop that technology within Dovetail, and then I helped our production lab implement that workflow in their day-to-day work. But I was, at the time, a little tired of having all my products axed by the commercial side of the organization. And so I kind of reached out to them and say, I understand there's not the market, but I want to understand why these decisions are being made the way that they are, and what can I do to help out. And so a few things that I did initially were to give lunch and learns. So whenever I was involved in a new technology, I would set up a meeting with our company and present what that technology was, how it worked, what it's good for, maybe what it's not so good for, things like that. And so between that and then having meetings with our sales team to help inform them and train them in the science a little bit better, they actually thought it was useful enough to actually bring me over to the commercial team, which is where I live now. So before actually doing that, kind of the skill sets that I had developed that made me really sort of desirable for moving into that role is fieldwork. I have spent so much time in the field that I understand the challenges that people go through. So when our customers are out there collecting samples for their genome assembly project, I can relate to them. I can say, oh yeah, I've been on a ship where the only thing I have to fix a sample is ethanol. This is how you would do that, right? And so I'm able to relate to the customers who live in the field and are doing experimental work. Wet lab, I understand how to pipette. I understand the nuances of doing molecular biology at the benchtop. And so again, I can relate to our customers that are going to buy our products and use them. because I've actually done that myself. And so I can walk them through where you're probably going to trip up, where you're not going to trip up, what are the little hints and tricks that make the process go better. And then bioinformatics. So this is something I didn't really talk about, but during my training, during my PhD, I was responsible for doing my own analyses. So I get all these data back and I have to make heads or tails of it. So I had to learn how to open up Terminal and basically learn to write in Linux and Python, and really learn how to use tools like R and MATLAB. And so I can really quickly now answer questions from customers instead of having to go to R&D or bioinformatics. I can just say, okay, where's your data? And I can open it up and I can ask the question. And so for customers that have problems, I can say, look, this is what the data actually are. And if you type this, this, and this into your command terminal, these are the answers that you're likely to get. And then you're outside of the parameters here, or you're within the parameters here. So this is a skill set that is actually really, really valuable. We lose an R&D bioinformatician to Google at least once a year, because genomics is big data. There are 3 billion bases in your genome, and we often sequence the genome to 30x coverage. So we're generating many billions of data points. And so if you can manage genomic data, you can manage any sort of tech data. So this is actually, if you're into computers and you're good with biology, this is a very, very lucrative route. I know undergrads that have been hired out of bioinformatics programs that make more than I do, and I have a PhD and they don't. It's a little annoying, but it's an incredibly valuable skill set, and it's actually one that, since I've been able to actually leverage to get a few raises here and there, be like, hey, I'm actually doing the computational work. You know, this would actually cost you about 10 times more if you were brought in somebody who's actually classically trained in informatics. I'm kind of self-taught, but I can get the job done. And another set of skill sets that is often not talked about in academia, but are incredibly, incredibly valuable. They're called soft skills. It's reading and summarizing literature. It's public speaking. I actually enjoy public speaking. It doesn't bother me. I know that weirds some people out, but it's something that I'm very comfortable doing. So being in front of customers or giving seminars, I travel the world now to give seminars at big universities on behalf of the company, or I present at conferences and things like that. I think it's fun. Some people think it's weird. Collaborations, being able to work with a big group of people and organize what are the questions, how are we going to address those questions, what are the deliverables that would actually answer those questions that we have set out that we define as important. Summarizing data, being able to make nice figures and take a really complicated data set and turning it into a simple image that people can understand. Teaching, part of going to grad school is being a TA at some point. I always had a blast teaching at Georgia Tech. I was actually one of the top ranked TAs out of all of Georgia Tech across any subject. And I thought that was hilarious because I never learned my students' names. I am terrible with names, but I always just made sure that everybody learned something and that felt comfortable in that environment, and they could ask whatever questions that they wanted. I didn't really care, how they actually did in the class. I just cared that they learned, and it ended up always being a good time, and everybody learned something and walked away with something valuable at the end of the day. So I found that very rewarding. Learning how to learn. So like I pointed out, my background is in oceanography and microbiology stuff. I am now doing human epigenetics stuff. I had to learn a lot about the human genome. I had to learn a lot about epigenetic stuff when I joined the company. It was tough, but part of getting your PhD is learning how to learn. And so that actually becomes quite a valuable skill set because our sales reps go, hey, here's this new technology. I don't understand how it works and how does it compare to ours. So I can teach myself how that technology works and then say, okay, these are the differentiators and this is how they're valuable, or where they're complementary and when they're actually competing or when they actually don't even jive at all. And the last one is storytelling with scientific data, right? that's really what brings people to the table. It's something that we've been doing since mankind had learned how to build fires, right? You sit around the campfire and you tell a story, right? That's what brings people together. And learning how to tell scientific stories that are not boring. I don't know, some of you might have thought this was boring. I don't know. But I generally find that being able to tell an interesting story keeps people engaged and often keeps that research alive and going. But all those soft skills combined with some of those hard skills that I gained has led me to my current role. So it's called scientific affairs. Really what it means is I'm a scientist for the commercial team. Oftentimes this means I am a translator from our research and development team into what would be useful into the market, and then vice versa. I can look at the market and translate for our R&D team and say how you should be spending your time, how you should be spending your few $1,000s of reagents and sequencing space to develop a assay or a kit that is going to be valuable to people in the field. And so my main tasks here are generating marketing material, presenting at conferences and seminars, solving very difficult technical questions for our customers, and then participate in core teams. So real briefly, I just wanted to walk you through maybe how this works in real life. So like I said, we make these sort of three-dimensional views of the genome. They're really expensive to make. You have to sequence about $20,000 worth of sequencing just to be able to build a contact map like this. People have been interested in what's called a chia pet or a high chip approach. We won't get into it. Basically, you enrich the data set for known sites before you sequence. Really saves you time, energy, and effort on sequencing, and it makes your analysis be quite straightforward. So instead of building this big thing, you want to be able to just look at what parts of the genome are interacting if you already know one region that you're interested in. So my company told me to go to this 3D genomics conference in Banff, Canada. If you ever get a chance, go. The place is absolutely stunning. This was the hotel that I stayed at. Work paid for everything. So I basically got to sleep in a castle in the mountains on my work's dollar, which was super cool. But during this time, I learned a lot. I went to the conference and I listened to the talks and I asked questions, and I figured out, I broke down the talks into what were the two main topics, how many I was actually able to attend. All of this market lived in the basic research. What applications were they using these data types to address? More specifically, what was that research area? Are they currently using similar technology to us? And do I think that they would actually buy from us? And so I brought this back into house and relayed that of this, one of the things that people were really interested in was this high chip, more streamlined approach to looking at genomic contacts. And they said, okay, cool, we'll keep that in the back of my mind, go out and look more. So I went out with our sales team to the field. So I hit Germany, England, Ireland, Switzerland, Stockholm, South Korea, Thailand, and Singapore, all within about 3 weeks. It was brutal. It was absolutely brutal. The travel was killer. But I saw a lot of the world very, very quickly. But I really went to help our sales team with seminars and to assist in one-on-one meetings. And during that, normal conversation, like, what are you guys interested in? And there were a number of different methods, but by and large, people wanted to hear about high chip, this protocol, high chip. So I was like, okay, that's great. I kind of had that suspicion. I have now confirmed that suspicion. So I bring that back to the company and say, hey, look, everybody wants to do high chip. Let's look at the market. Well, this is the market of ChIP-seq. This is the market of Hi-C. This is where I think the product would eventually end up making. These are the sample types that we see in the publications, and these are the most common antibodies used in this protocol. So if we're going to make this protocol, this is the set that we should be, incorporating into our product. And then, we just launched HiChip at a big major conference at AGBT right before they shut down the planet, basically. And this particular figure was a figure that I made where you're looking at ChIP-seq data, and then here's HiChip data where you can see the interactomes between these peaks. And this particular image has already sold us about $250,000, and we literally just launched this at the end of last month. So it's really being able to distill that information into an image that people want to see and a product that people want to buy. So like we launched this at end of February. We sold the first product less than a week later. So that's kind of one of the many aspects of my job that I do is I go out in the field, I hear what people are doing, I look at the science, what are people doing in science, how are people publishing, and what do I think will resonate with them? And then I take that and bring it back to our R&D team and our commercial team and say, this is something that's viable. Let's do the work, figure out if it's worth doing, and then, go for it. So this started pretty much a year ago when I went to Canada for a conference. And within a year, we had launched a product that people actually really want to buy. So just closing thoughts, Nick, which Nick made me articulate much earlier, is you have to talk to people. You have to make friends, be helpful, be positive, get excited. Even if that's not in your nature to be that excitable of a person, it helps. When you're interested in something and people can see that, they are really willing to help you understand everything about that particular thing. Very few things are achieved alone. This is just a very small smattering of the people that have been really influential in not only my education, but in me being able to get a job. I learned a lot of just like task tracker stuff and day-to-day organization and how you report to a boss in different types of jobs that, like I don't think, actually I know I would not be here if not for these people. Just hands down, I wouldn't be here talking to you guys right now. I'd be working in an aquarium shop. And be open to opportunities. You never really know where they're going to take you. Like I said, I didn't really want to do the senior thesis, but I did. And that led to a snowball moving downhill, which led to me getting a Ph.D., which led to me getting a biotech job, which lets me kind of travel the world. And then I also work from home the rest of the time. So I get paid a decent salary. I get to travel. I get to work from home, and I get to still be involved in really cool science. And always ask questions. If you don't get it, it's up to you to vocalize that. And I used to tell students this in my TA class that I TA is, right now, your education probably is costing you between $100 to $300 per class, at least when you go to bigger institutions. And so if at the end of every class, if you don't feel like you got your money's worth, you should be speaking up and making sure that you got what you wanted out of that class. The education part is really up to you. Teachers will teach what's in the textbook, but if you want to know something that's not in there, you got to speak up. You got to make sure you get your most the bang for your buck there, because that's coming out of your pocket or your parents' pocket at the end of the day. And it's really it's on you to learn. And so if you don't feel like you got the knowledge that you wanted, say something. And that's it.
All right. Does anyone else have any questions or anything? I know I may start this off. Cory, actually, your business analysis looked pretty impressive on there. I mean, what did you?
So that I've actually been our VP of commercial operations has now made it mandatory that everybody follows my analysis workflow when they go to conferences, or even when they're out in the field. And so when you're doing your scientific training, right, a lot of it comes down to data analysis. You can do all the experiments that you want to do, but if you don't have an analysis in place, you're never going to be able to answer your question. And so really, I really credit my PhD advisor for this. Him and I would argue ad nauseum about, is a figure better or is a table better? And now that I'm a bit older and more experienced, a well-organized table is the best approach to any sort of technical problem in the world. You can ask many questions from it. You can set it up a variety of different ways. You can distill it down a variety of different ways. It's really just, how do you set yourself up ahead of time? Let's see, give me 1 second. I can actually pull up that particular, if you guys can take a look, it's really just, setting up a table and knowing that you're going to say what percentage of people did what, and what questions do you want to get out of it. Let's see. So you guys can keep asking questions while I find this out.
Well, it seems like you made a change from being kind of just the research scientist to being tired of having your project shot down by business, and that you went into the business. Are you still getting that kind of same, where people who do research are a special lot, and are you still getting the same satisfaction out of going more on the business side?
So real quick, this is just how I set up my tables and stuff like that. But yeah, that is a great question. I kind of always knew at the end of the day that I enjoyed storytelling way more than I actually enjoyed being in the bench or out in the field. It's one of those things, you do all this hard work, and so telling the story at the end of the day, how you got there, that's the fun part. And so, having an opportunity to look at the business side of thing, one, as a scientist, it gives me a different view of how companies are run. I can bring my analytical brain to their side of the business and say, you might have a gut feeling that this might be better, but let's prove that that's the better thing. So I'm still quite satisfied in how much I'm engaged with science on the commercial side. There are a few things that just really drive me up the wall from time to time, which is like right now, they're really pushing for a social media platform stuff. And personally, my philosophy on this is I hate the fact that social media exists. I despise the fact that Twitter is a thing. But anyway, they're wanting me to get involved in that, and I'm saying that's not science. that's societal vomit. But whatever, I will generate the scientific content for you guys to put up there, but I'm not going to manage that. Right? That has to go to somebody else because I can't. I would lose it. A part of me died inside when they brought up that conversation. But I do because I have so many sort of skill sets in terms of the informatics and the storytelling. It's really funny. Both R&D and the informatic team gives me tables at the end of the day and says, "Can you make this into a figure for our tech notes?" And the answer is yeah. A lot of the time I can. So it's kind of nice in that I don't have to do a lot of the grunt work to make those tables. Or I tell them how I want that table structured, and then they go give me the data. And then I can go make those really cool images and that relate to the science. So the other thing that, I kind of go back and forth on is dealing with the sales team. I did not get a PhD to sell stuff. I got a PhD to do sciences. But you do have to realize that when you walk away from academia, right, and going into the business world, that the dollar is kind of the bottom line. And your sales team, who may or may not be technically trained, are the tool by which you bring in that bottom line. And so you do your best in training them as much as you can, but there are many cases where I get pulled into customer conversations where I have bigger fish to fry. They're trying to sell 1 or 2 kits, which is a few $1,000s, where I'm trying to address something that could probably bring in multiple millions of dollars. So trying to find that balance of, I don't need to be in this one-off meeting that might help you sell one thing versus being in a position where I'm helping develop a product that could be, several to tens of millions worth of revenue for a company. I do feel like I still get the science. I still get my science rocks off, but it's it's instead of driving towards publication, it's driving towards a product, which in biotech you feel like they should be one and the same thing, but sometimes that doesn't always hold true. So, for example, that Oxford Nanopore company I was telling you about, really amazing technology, but they're probably 5 to 10 years off of being an actual viable product. But everybody wants us to make our DNA data approachable through their platform. We actually don't think their platform is the best sequencing platform to answer our question, but the market wants that product. So do you go against your better science nature and say, that's actually not the right approach here, you should do it this way, versus saying, actually, they're going to buy this no matter what we do, we might as well just make it. So I still don't know how to navigate those waters yet, because I'm still fairly young in biotech, and particularly even younger on the commercial side or on the business side. So things like that kind of test my scientific background a little bit. But at the bottom line, the company's got to make money, so I can have a job and have health insurance and all that fun stuff.
All right, great. So, so this is the table that you have on the. You put up a table, and then you make the. So you made some really great figures off of that table. So you're
so then I take this, turn it into a comma separated table, import it into R, and then I tell it to summarize. I summarize all my columns. How many variables are in each column? What's the count? What's the percentage of those? So that can be pretty straightforward. You can get a little bit more complicated when you want to draw Venn diagrams to say how many people are using this technology plus this technology, right? How many people are interested in gene regulation and development? How many people are interested in cancer and transcription? So you can play those games as much as you want, and doing those sort of analyses can be a bit more complicated, but there are tons of computational packages that if you just set up a good table, you can answer them all.
Okay. And so you would suggest that students do learn how to use Excel, use the power of Excel or Access as much as they can.
Oh yeah, so I'll pull something else up here. That's a really good question. So actually, my day-to-day operations are run, and my boss tracks what I do through this. It's just a task tracker, right? This is how I organize. Here's the category of things that I'm working on. Here are the particular descriptions of the tasks that I have to do. Here's the priority in which I rank them. What do I actually need to finish these things? What's the due date? And then update it as I go along. As things get completed, I toss them into this. I track my travel. I even track my company-wide goals that I have set out for myself. These are useful tools, and it's funny now that we've all moved to remote. Our CEO is asking for everybody to submit task trackers to their supervisors. And it's funny, our supervisor circulated this, or my task tracker, around to the company as a guide of how you should be setting up your task trackers. It really, I cannot understate the value of a good table. It can answer every question that someone. So right now, if someone says, how many hours do you need to finish up all these projects? Well, you can just sum this column and say, I need 80 hours at minimum to be able to answer all of these questions, or to close off all of these tasks, right? And so if you want to give me any new tasks within the next week, what on this task tracker falls off?
Yeah.
Right. It becomes it's a very easy way to say, okay, here are my checks and balances. Here are my resources. Here are resources that I need. It becomes a much easier way to manage and ask for those things. And then, bioinformatics is all about making tables, right? You can't do analyses without the tables to ask the questions. So yeah, be good with your tables. It goes a long way. You're never going to go away from them. Even things like HubSpot, if people are really interested in going to the business side, HubSpot and Salesforce, just basically turns a form that you fill out for each customer or for each campaign, and it turns that into a table, and then your dashboards are built from that table. So if you have a bar graph or a pie chart on your dashboard, the only way you get that is for HubSpot and Salesforce to turn that into a table to be able to ask questions of the data. Right? At the end of the day, science is all about generating the data and then asking your questions of that data. And so you have to organize the means or the media by which you ask those questions. And in sciences, it's tables.
Okay, so Cory, we're a little bit over time and stuff like that, but we have about 9 participants. I think at the height we had about 20, so that's good. I will I videotape this, so I'll make this available to our students and stuff. And so a number of our students say thanks. So I will see you after this this time gets over, and we'll be able to play some water polo, and I'll take you out for a beer. So thank you very much for doing this, Cory.
Sounds good. Yeah, I'm jonesing to score some goals here. I'm kind of going. I've been playing hockey on my Xbox like crazy just to get the satisfaction of scoring on a goalie.
Okay. All right.
All right. Sounds good.
We'll catch you later.
All right.
Bye now.
Bye.